Welcome back to the Deep Dive. We're going straight into, well, one of the most intellectually compelling and, let's be honest, frustrating challenges in modern medicine, the complex, really nuanced mechanisms of autoimmunity.
Yeah.
Today's Deep Dive is all about that delicate balance of the immune system and what happens when it just loses its internal compass, the essential principles of self-attack.
It's such a critical topic. I mean, we're talking about the body attacking itself, often with, devastating chronic consequences.
Right.
And for many listeners, this probably isn't abstract at all. Autoimmunity, it's estimated to affect a pretty significant chunk of the U.S. population, somewhere between, say, 5% and 10%.
Wow, that's substantial.
It really is. And maybe more concerning, we're seeing the incidence rise, especially in high-income countries. That really suggests a strong interaction between, well, our genes and our modern environment.
Yeah, that interaction seems key.
Yeah.
Now, when we talk about autoimmunity, it's easy to just sort of wave a hand and say, oh, the body's confused. But true pathological autoimmunity, the kind that causes disease, it actually requires a very specific set of failures, doesn't it? Because, I mean, you check the sources, and even perfectly normal, older individuals can sometimes have these autoantibodies just floating around without ever getting sick.
That's the crucial distinction, absolutely. The sources, they lay out 3 core requirements for classifying a disorder as, truly pathological autoimmunity.
Okay, what are they?
So first, you absolutely must have an immune reaction that's specific for a self-antigen or a self-tissue. Got to be targeted itself.
Makes sense.
Second, there needs to be compelling evidence that this specific reaction is the primary cause of the damage. It can't just be, say, secondary cleanup after the tissue has already decayed for some other reason.
Right, it has to be the driver.
Exactly, the driver. And third, you have to rule out any other well-defined cause. It really must be a primary breakdown of self-tolerance.
Okay, so it's quite specific. Let's maybe establish the clinical spectrum we're dealing with here, because it's not just one thing, is it?
Not at all. It's a huge range. On one end, you have the highly targeted, almost surgically precise, organ-specific diseases. Think type 1 diabetes, for example.
Right, where the attack just wipes out the insulin-producing beta cells in the pancreas. Nothing else.
Precisely. Or multiple sclerosis, which is laser-focused on the myelin sheaths wrapping around nerves in the central nervous system. Very specific targets.
Okay.
But then, at the other extreme, you have these widespread systemic diseases where it feels like the immune system is just throwing everything it has at the body all at once.
Like lupus.
Systemic lupus erythematosus, SLE, yes, that's the classic systemic example. In SLE, the antibodies are directed against antigens that are found everywhere, nuclear DNA, platelets, blood cells.
So it hits multiple systems.
Exactly, leading to really complex multi-organ problems involving the kidneys, the joints, the skin, you name it. Basically, the nature of the target dictates the kind of pathology you see.
So if the whole system is fundamentally designed to live in harmony with our own cells, what you called self-tolerance, then our core mission today has got to be figuring out exactly how that profound and really elegant system breaks down.
Definitely. And to understand the failure, the breakdown, we first have to really get a handle on the success. How does it normally work?
Right.
Self-tolerance is achieved through something called immunologic tolerance. Basically, it's unresponsiveness to an antigen induced by being exposed to it. And the system operates through 2 critical, quite complex lines of defense. Think of them like layers of security clearance: central tolerance and peripheral tolerance. Okay, let's start with that first layer. Central tolerance. This acts as the primary filter, the main screening process.
Where does this happen?
It occurs right in the central lymphoid organs. So that's the thymus for T cells and the bone marrow for B cells.
Okay.
And the goal here is, well, it's simple but kind of brutal. Eliminate or maybe reform any potentially self-reactive lymphocyte clone before it ever gets out into circulation. Stop it at the source.
So for T cells in the thymus, how does that work? I think you mentioned negative selection.
Yeah, negative selection, or sometimes called clonal deletion. Basically, immature T cells arrive in the thymus, and they have to pass this really stringent test.
Okay.
If their newly created T cell receptor, the thing that recognizes antigens, happens to bind too strongly to a self-antigen being presented by cells in the thymus,
No.
then that T cell is programmed to die. It undergoes apoptosis. It's like boot camp. Only the compliant soldiers, the ones that don't react dangerously to self, are allowed to graduate.
But wait, how can the thymus possibly show these developing T cells all the potential self-antigens? I mean, what about proteins that are normally only found in, say, the pancreas or the liver?
Ah, that is a fantastic question. And the body has this brilliant solution called the AIR protein. AIR stands for autoimmune regulator.
Autoimmune regulator. Sounds important.
It's absolutely critical. What AIR does is, well, it essentially turns certain cells within the thymus, these medullary epithelial cells, into a kind of comprehensive molecular sampling committee.
How does it do that?
AIR actually induces these thymic cells to express hundreds, maybe even thousands of antigens that are normally restricted only to peripheral tissues, things you'd usually only find in the liver or the kidney or the pancreas, like you said.
Wow. So the thymus basically creates a library of self-tissues just for testing.
Pretty much. It's this massive quality assurance check. It forces the developing T cells to be tested against a huge range of self-antigens they wouldn't otherwise encounter until they leave the thymus and go out into the body.
That's incredible. So the body literally sacrifices the normal cell identity of some thymic cells just for safety screening. What happens if this AIR switch is faulty?
The consequences are severe and very direct. There are rare cases of people born with germline loss-of-function mutations in the AIR gene.
And they get.
They develop something called autoimmune polyendocrine syndrome, or APS1. Because the T cells skip that crucial screening step for peripheral antigens, they enter the circulation.
Still potentially self-reactive.
Exactly. And they then systematically attack and destroy multiple endocrine organs, the parathyroids, the adrenals, the pancreas. It tragically proves just how vital AIR's role is in establishing central tolerance.
Okay, so AIR is key for T cells. Now, you mentioned that if a T cell sees self-antigen in the thymus, it either dies or there was an alternative.
Right, or it can be diverted. Instead of dying, some T cells that recognize self-antigens with intermediate strength can actually differentiate into a special type of cell called a regulatory T cell, or TREG. We'll definitely come back to TREGs when we talk about peripheral tolerance.
Okay, TREGs. Got it. Now, let's switch over to B cells. They mature in the bone marrow. Do they have AIR too?
No, V cells don't use AIR. They have a different strategy, which is actually less lethal initially. It's called receptor editing.
Receptor editing sounds like they get a do-over.
That's a great way to put it. B cells are essentially given a mechanism for redemption. If a developing B cell's receptor strongly recognizes a self-antigen in the bone marrow...
Instead of just dying...
Exactly. Instead of immediate apoptosis, it reactivates the machinery that rearranges the antibody genes, the same process that created the receptor in the first place, VDJ recombination.
So it tries again.
It tries again. It attempts to essentially cut out the self-reactive gene segments and splice in new ones, expressing a brand new antigen receptor that is hopefully not self-specific.
They get a chance to rewrite their own dangerous potential. That sounds like a pretty complex process. Does it happen often?
Oh, extremely often. Current estimates suggest that somewhere between maybe 25% and up to 50% of all maturing B cells actually undergo receptor editing.
Wow, half of them.
Potentially, yeah. It just shows the incredible lengths the system goes to ensure self-tolerance. It's a huge investment.
Yeah.
Only if receptor editing fails or if the B cell just can't manage to create a non-self-reactive receptor after trying, then it undergoes apoptosis in the bone marrow.
Okay. So we have these amazing multi-stage filters, negative selection for T cells, ARE, making sure they see lots of stuff, receptor editing for B cells. But you mentioned these systems aren't perfect.
No, they're not. Central tolerance is inherently imperfect. It's just not possible for every conceivable self-antigen to be present at the right concentration in the thymus or the bone marrow during development.
So inevitably.
Inevitably, some self-reactive lymphocytes are going to escape into the periphery. They slip through the net.
And that's where the second layer of defense kicks in.
Exactly. This is where peripheral tolerance becomes absolutely essential. It's the safety net operating out in the tissues. Hashtag tag 1.2. Peripheral tolerance, the secondary filter. So peripheral tolerance is basically the continuous patrol system. It's designed to silence or eliminate any autoreactive lymphocytes that manage to slip past that central filter in the thymus or bone marrow.
Okay, how does it do that? What are the mechanisms out in the field?
There are several key ones. The first critical mechanism is inducing anergy, which basically means functional unresponsiveness.
Anergy.
Okay.
We often hear it simplified as paralysis for the cell, but what's actually happening at the molecular level to cause that?
Yeah, paralysis is a good shorthand. It connects directly back to that fundamental rule of T cell activation. the two-signal requirement.
Right, you need more than just seeing the antigen.
Absolutely. For a T cell to get fully activated and ready to fight, it needs signal 1, which is its T cell receptor recognizing the antigen peptide presented on an MHC molecule.
Okay.
And it needs signal 2, which comes from co-stimulatory molecules on the antigen-presenting cell, APC, typically the B7 molecules, B71CD80 and B72CD86, binding to the CD28 receptor on the T cell.
Signal 1 is what to attack, signal 2 is permission to attack.
That's a perfect analogy. Signal 2 is the go signal, often indicating danger or inflammation. Now think about normal, healthy tissues. The main APCs there are often resting dendritic cells or other tissue cells. Crucially, these resting cells typically lack sufficient levels of those B7 co-stimulators.
So they're showing self-antigens but without the go signal.
Precisely. So an autoreactive T cell wandering through that tissue might see its self-antigen presented, signal 1, but because the APC isn't signaling danger, that crucial signal 2 is missing or very weak.
And what happens to the T cell then? Does it just ignore it?
It's worse than ignoring it. Getting signal 1 without adequate signal 2 doesn't just result in inaction, it actively triggers a negative outcome for the T cell. The cell actually engages inhibitory pathways. It might upregulate inhibitory receptors. It can degrade necessary signaling molecules.
So it gets shut down.
It gets shut down hard. It enters this state of profound and persistent functional paralysis that's anergy. It's essentially been reprogrammed to ignore that specific self-antigen from then on, even if it encounters it later with co-stimulation.
Wow. So encountering self in a safe context induces paralysis. Is this paralysis reinforced?
Yes, definitely. It's constantly reinforced by other molecular inhibitory signals. These are like the immune system's natural brakes. They're crucial for maintaining peripheral tolerance.
Yeah, and the big names here are?
The major players, the ones getting a lot of attention now, especially in cancer therapy, are the co-inhibitory receptors, CTLA-4 and PD-1.
Okay, CTLA-4 and PD-1, how do they work?
So CTLA-4 is structurally very similar to the activating receptor CD28. In fact, it binds to the same B7 molecules on APCs that CD28 binds to.
But it sends the opposite signal.
Exactly. It competes with CD28 for B7 binding, but when CTLA-4 engages B7, it delivers a powerful off signal to the T cell, actively shutting down activation.
And PD-1?
PD-1 works a bit differently. It binds to its ligands, PDL1 and PDL2, which can be expressed on APCs, but also on many other tissue cells and even tumor cells. PD-1 engagement also delivers inhibitory signals, and it seems particularly important in shutting down T cells that are chronically stimulated, like in persistent infections or potentially self-antigen exposure.
The clinical proof for these being breaks is pretty dramatic, isn't it?
Absolutely.
Yeah.
If you genetically knock out the genes for CTLA-4 or PD-1 in mice, they spontaneously develop severe, widespread systemic inflammatory diseases and autoimmunity. And similarly, humans who inherit rare genetic defects in these pathways also suffer from serious autoimmune conditions. It really underscores that these molecules are absolutely essential for actively suppressing self-reactivity in the periphery. They are the brakes.
Okay, so anergy and inhibitory receptors are key. What's next?
Well, perhaps the most crucial active regulators out in the periphery are the regulatory T cells, TREGs we mentioned earlier.
Right, the ones that can develop in the thymus.
Yes, some develop there, natural TREGs, and others can be induced in the periphery too, induced TREGs. These cells are the master coordinators of non-response, the peacekeepers of the immune system.
How do we identify them? What are their markers?
They are typically CD4 plus T cells, so helper T cell lineage. Crucially, they express high levels of the IL-2 receptor alpha chain, which is CD25. But the absolute defining marker, the master switch, is the transcription factor FOXP3.
FOXP3.
Yeah.
What makes FOXP3 so central to what TREGs do?
FOXP3 is basically the master transcription factor that dictates the entire TREG developmental program and function. Think of it like the central operating system for suppression.
Okay.
It directs the TREG to produce and secrete powerful immunosuppressive signaling molecules, cytokines, mainly interleukin-10, IL-10, and transforming growth factor beta, TGFM.
And what do those cytokines do?
They act on the surrounding cells. They actively inhibit the activation, the proliferation, and the effector functions like killing or cytokine production of other nearby T cells, and also antigen-presenting cells. So TREGs aren't just passively unresponsive, they are actively suppressing the potential immune battle around them.
They're enforcing the peace.
Exactly. And once again, the proof of their importance comes from what happens when they fail.
Let me guess, severe autoimmunity.
You got it. Mutations in the human FOXP3 gene cause a devastating devastating X-linked syndrome called IPEX.
IPEX.
Stands for immune dysregulation, polyendocrinopathy, and neuropathy, X-linked syndrome. It's a severe multi-system autoimmune disorder that often presents very early in life, usually fatal if not treated aggressively, often with bone marrow transplant.
Wow, so losing just this one transcription factor, FOXP3, causes the whole peripheral tolerance system to essentially collapse.
It demonstrates just how critical that single coordinator is. Without functional TREGs, you get disastrous, unconstrained self-attack across multiple organ systems. It's a tragic but powerful lesson in TREG function.
And it's interesting, the source notes remind us TREGs aren't just policing our own tissues against autoimmunity, right?
That's a really important point. TREGs are also essential for maintaining tolerance in other contexts. For example, preventing the mother's immune system from rejecting the fetus during pregnancy. Remember, the fetus expresses paternal antigens that are foreign to the mother, and they're crucial for maintaining immunologic peace with the trillions of commensal microbes living in our gut. We don't want to attack those beneficial bacteria. TREGs help maintain that tolerance. They really are global harmony keepers.
Okay, so we have energy, inhibitory receptors, and active suppression by TREGs. Is there a more definitive way to deal with escaped self-reactive cells?
Yes. The third essential mechanism is just outright dilution by apoptosis, killing off the self-recognizing cells out in the field.
Programmed cell death again. How is this triggered in the periphery?
There are a couple of major pathways. One involves the intrinsic or mitochondrial pathway of apoptosis. Self-reactive T cells that keep seeing their antigen without the right context might upregulate pro-apoptotic proteins like BIM, while failing to upregulate the protective anti-apoptotic proteins like BCL2 or BCL-XL. This imbalance tips the scales towards cell death.
Okay, the cell basically self-destructs. What's the other pathway?
The other major one is the extrinsic pathway, mediated by death receptors. The most famous pair here is FOS, also called CD95, and its binding partner Fas ligand, FASL.
How does that work?
When lymphocytes, particularly T cells, are repeatedly stimulated by self-antigen, they tend to upregulate the expression of the phase receptor on their surface. Activated T cells can also express Fas ligand.
So they can trigger death in each other.
Exactly. When Fas allele on one cell binds to Fas on another, or even potentially on the same cell autocrine signaling, it triggers a rapid signaling cascade inside the Fas-expressing cell that leads directly to apoptosis. It's often called activation-induced cell death.
And do we have human proof for this pathway's importance in tolerance?
We do. Similar to CTLA4 and FOXP3, inherited mutations in the genes for either Fas or Fasl cause a human disease called autoimmune lymphoproliferative syndrome, or ALPS.
ALPS, what does that look like?
People with ALPS have difficulty deleting lymphocytes, so they accumulate massive numbers of them, leading to swollen lymph nodes and spleen. And importantly, they develop autoimmune reactions, particularly against blood cells, and have features sometimes reminiscent of lupus, SLE. It shows that failure of this death pathway directly contributes to autoimmunity.
Okay. One last mechanism mentioned is antigen sequestration. This sounds more passive.
It is more passive, but still effective. It refers to the fact that certain sites in the body are considered immune privileged. Think of the testes, the inside of the eye, and the brain.
What makes them privileged?
Primarily, antigens within these sites are physically hidden or sequestered from the main highways of the immune system. They often have barriers that limit the entry of lymphocytes and antibodies, and they don't drain readily into the lymphatic system where immune responses usually get initiated.
So the immune system just doesn't normally see the antigens inside.
Largely, yes. It's like keeping highly reactive materials behind a thick shield. The downside is, if these sequestered antigens are suddenly released maybe due to major trauma, like a penetrating eye injury or a severe infection,
Then the immune system sees them for the first time.
Exactly. And because the immune system was never educated to tolerate these antigens during development, they were hidden, it treats them as completely foreign. The resulting immune response, like the post-traumatic uveitis you can get after an eye injury, can be incredibly severe and prolonged. It's inflammation triggered by the release of previously hidden self.
Okay, we've laid out this really complex, multi-layered system of tolerance, central and peripheral checks and balances. The picture that emerges is one of a system that's incredibly robust in many ways, but also maybe surprisingly fragile at key points.
That's a good way to put it, robust yet fragile.
So let's flip the coin now and really dig into the breakdown. How does autoimmunity actually happen? The sources seem very clear that it's rarely just one single defect causing the whole problem.
Almost never. It's typically portrayed as this devastating synergy, a combination of factors coming together.
What are the main components of that synergy?
It boils down to two major factors. First, underlying susceptibility genes that somehow compromise those tolerance mechanisms we just discussed. They create the vulnerability.
The genetic predisposition.
Right. And second, environmental triggers, things like infections or tissue injury or even UV light that come along and essentially activate the compromised system, pushing it over the edge into self-attack. You generally need both. Hashtag 2.1. Role of susceptibility genes, the setup. That synergy is absolutely vital to understand. Autoimmune disorders are fundamentally complex, multigenic disorders. The strongest evidence for the genetic component comes from twin studies.
How so?
Well, if you look at identical twins who share 100% of their genes, their concordance rate for developing a specific autoimmune disease, meaning if one twin has it, the other also gets it, is significantly higher than for non-identical or fraternal twins who only share about 50% of their genes.
So genetics clearly plays a big role.
A huge role. It provides that critical underlying vulnerability. But it's not the whole story, because even in identical twins, the concordance is never 100%. If one identical twin has lupus, the other might only have a 25% chance, roughly, of getting it.
Which tells you environment matters too.
Precisely. Genes load the gun, environment pulls the trigger. Now, when we talk about the genes involved, the single most critical genetic contribution, time and time again, comes back to the HLA genes.
HLA, the human leukocyte antigen genes, these encode the MHC molecules, right? The ones that present peptides to T cells.
Yeah.
Why are they so dominant in autoimmunity risk?
It really comes down to their function. MHC molecules are the display platforms. They determine which peptide fragments from both foreign proteins and our own self-proteins get presented to T cells.
Okay.
So the thinking is that certain specific HLA alleles, different versions of these genes, might be particularly good or maybe particularly bad, depending on your perspective, at binding and displaying certain self-peptides.
Meaning they might show a self-peptide in a way that looks dangerously foreign to a T cell?
Exactly. Or perhaps they fail to effectively present certain self-peptides in the thymus during negative selection, allowing potentially dangerous T cells to escape. Or maybe they're really good at presenting peptides derived from infectious agents that happen to mimic self-peptides. We'll get to mimicry later. The exact mechanism can vary, but the core idea is that your specific HLA type profoundly influences which peptides your T cells see and react to.
And the risk associated with certain HLA types can be pretty dramatic.
Oh, absolutely striking. The classic example is ankylosing spondylitis, a type of inflammatory arthritis primarily affecting the spine. It has an incredibly strong association with one specific HLA allele, HLA-B27.
How strong?
If you inherit HLA-B27, your relative risk of developing ankylosing spondylitis jumps by something like 100 to 200-fold compared to someone without it.
Wow. That's an enormous risk multiplier.
It is. It really demonstrates how profoundly the specific way antigens are presented can influence self-tolerance. But, and this is the absolutely essential point we need to keep emphasizing.
Let me guess. Having HLA-B27 doesn't guarantee you'll get the disease.
Not even close. The vast majority of people walking around who carry the HLA-B27 allele never develop ankylosing spondylitis.
So it's a major risk factor, but not deterministic.
Exactly. The HLA genes set the stage. They provide the susceptibility, perhaps making the immune system capable of making a mistake, but they're clearly insufficient on their own. You need other factors, likely other susceptibility genes and those critical environmental triggers to actually launch the autoimmune attack.
Okay, so HLA is the big one. But modern genetics, like genome-wide association studies, GWAS, they found other genes too, right? Non-MHC genes.
Yes, absolutely. GWAS has been really powerful here. It's revealed a whole host of non-MHC genes that contribute smaller amounts of risk, but collectively are very important. And interestingly, many of these genes are involved in general immune regulation pathways.
Can you give some examples? What do these genes do?
Sure. One of the most frequently implicated non-HLA genes across several autoimmune diseases, including rheumatoid arthritis and type 1 diabetes, is PTPN22.
PTPN22. What does that code for?
It encodes a protein called lymphoid tyrosine phosphatase, or LYP. As a phosphatase, its job is generally to remove phosphate groups from other proteins. In T cells, it acts as a negative regulator. It helps to dampen down the signaling that occurs right after the T cell receptor gets engaged. It's a break.
Okay, so it turns signaling off, and the disease-associated variants.
The common disease-associated variant of PTPN22 is thought to be a gain-of-function variant in its phosphatase activity, paradoxically.
Wait, stronger braking leads to autoimmunity. That seems backward.
It does seem counterintuitive at first, but the current thinking is more nuanced. This hyperactive phosphatase might too effectively dampen signals coming through the T-cell receptor during development in the thymus.
Ah, so it might interfere with negative selection.
Possibly. Or perhaps by dampening weak signals from self-peptides, it prevents the induction of tolerance or energy in the periphery. Alternatively, it might interfere with TREG function. The precise mechanism is still debated, but the net result seems to be that T cells become less well-regulated and potentially more prone to activation against self-antigens, leading to excessive lymphocyte activity.
Okay, so PTPN22 affects T-cell signaling thresholds. What else?
Another really interesting one is NOD2. Polymorphisms in NOD2 are very strongly associated with Crohn's disease, which is a major form of inflammatory bowel disease, IBD.
Crohn's. And what does NOD2 do?
NOD2 is fascinating. It's an intracellular sensor, primarily found in immune cells and also the epithelial cells lining the gut. Its job is to detect specific components of bacterial cell walls, particularly muromyl dipeptide. It's part of the innate immune system's surveillance for microbes.
So it senses bacteria. How does a defect lead to Crohn's?
The common disease-associated variants of NOD2 are actually loss of function. They make the protein defective in sensing these bacterial components.
So the immune system doesn't see the bacteria properly. How does that cause inflammation?
Again, a bit counterintuitive. One leading hypothesis is that in the gut, NOD2 plays a crucial role in maintaining tolerance to our normal commensal gut microbiota. By properly sensing these bacteria, it might normally trigger responses that keep them in check or induce tolerance. If NOD2 is defective,
It fails to send the all-clear or normal residence signal.
exactly. The innate immune system might fail to properly recognize and tolerate these normal gut inhabitants. This could lead to an overreaction by other parts of the immune system, mounting a chronic, inappropriate inflammatory response against bacteria that should be ignored, and that inflammation then spills over, causing damage to the gut tissue itself.
So in this case, the primary issue might be a failure to tolerate something non-self, commensal bacteria rather than self, but the collateral damage hits self-tissue.
That's a very plausible model for Crohn's, yes. It highlights how complex the interactions are.
Are there others? You mentioned general immunoregulation.
Yes, we also find polymorphisms in genes that are absolutely core to immune balance. For example, genes involved in the IL-2 signaling pathway. IL-2 is critical for the development and survival of those regulatory T cells, the TREGs.
Ah, so defects there would impair TREG function.
Precisely. Polymorphisms in the gene for the IL-2 receptor alpha chain, CD25, or even IL-2 itself, are linked to diseases like multiple sclerosis and type 1 diabetes. If your TREGs aren't getting the right signals to develop or function properly, your peripheral tolerance is weakened.
Makes sense.
Similarly, polymorphisms in the gene for CTLA4, that inhibitory receptor we discussed, are also associated with several autoimmune diseases like type 1 diabetes and Graves' disease. Again, if one of your key immune breaks is genetically a bit weaker, you're more susceptible to losing control.
So the picture from the non-MHC genes is really about disrupting that fundamental balance between activation and inhibition, between effector cells and regulatory cells.
That's exactly it. The genetics often tips the scales, making the whole immune economy more prone to instability and self-attack. Hashtag CheckChat 2.2, role of infections and environmental factors. The trigger.
Okay, so the genes set the stage, they compromise the tolerance mechanisms, they load the gun. But you said that's usually not enough. You need the environment to pull the trigger.
Correct. And the most well-studied environmental triggers are infections, but tissue injury and other factors play a role too. These events seem to provide the necessary spark to ignite autoimmunity in a genetically susceptible person.
How does an infection, which is supposed to trigger an immune response against a pathogen, end up causing an attack against self? What are the mechanisms?
There are two main mechanisms proposed, and they're not mutually exclusive. Both could be happening. The first is all about breaking that state of T-cell energy we talked about. Let's call it mechanism A, upregulation of co-stimulator.
Okay, breaking energy by boosting signal 2.
Precisely. Imagine you have a local infection, say, in your joints or your thyroid gland, or maybe significant tissue injury occurs there. This immediately triggers an innate immune response in that local area. The alarm bells go off.
Right.
As part of that innate response, local antigen-presenting cells, APCs, like dendritic cells and macrophages, become activated, a key feature of this activation is that they dramatically increase their expression of those crucial costimulatory molecules on their surface, B7-1 and B7-2. They start waving the danger flags.
So the local environment becomes inflammatory.
Highly inflammatory, yes. Now imagine one of these highly activated ATCs, loaded with costimulators, happens to pick up and present a self-antigen from the local tissue, maybe just as bystander uptake during the inflammation.
Okay.
Now along comes that poor, previously anergic T cell, the one that recognized this self-antigen before but got signal 1 without signal 2 and was paralyzed. This time, however, it sees its self-antigen, signal 1, presented by an APC that is also shouting danger with lots of B7, signal 2.
So now it gets both signals.
It gets both signals loud and clear. And this combination is enough to break the paralysis of anergy. The self-reactive T cell becomes fully activated, starts proliferating, and initiates an attack against the tissue displaying that self-antigen. The infection essentially provided the missing costimulatory fuel to ignite the pre-existing but dormant self-reactivity.
The susceptibility was there. The infection provided the spark. Okay, that makes sense. What's the second mechanism? You mentioned mimicry.
Right. Mechanism B, molecular mimicry. This one is perhaps more direct, but also maybe more insidious. It happens when a microbe, a bacterium or a virus, happens to express proteins or other antigens that bear a strong structural resemblance, often at the amino acid sequence level, to one of our own self-antigens.
So the pathogen looks like self by coincidence.
By unlucky coincidence, yes. The immune system, naturally, mounts a vigorous and entirely appropriate response against the foreign invading microbe. It generates T cells and antibodies specifically targeting the microbial antigens. But because of that structural similarity, some of the activated lymphocytes or antibodies produced against the microbe also recognize and react with the similar-looking self-antigen. They cross-react.
The immune system gets confused because of the resemblance and attacks the wrong target.
Essentially, yes. It mistakes the self-protein for the pathogen it was originally fighting. The absolute classic textbook example of this is rheumatic heart disease.
How does that work?
Rheumatic fever and subsequent heart disease can develop after an infection with certain strains of Streptococcus bacteria, the ones that cause strep throat.
Okay.
It turns out that proteins in the M protein family on the surface of these streptococci share structural similarities, specific amino acid sequences, with proteins found in human heart muscle, the myocardium, and heart valves.
So antibodies made against the strep bacteria.
And binding to the heart tissue as well, triggering inflammation, myocarditis, and over time, potentially damaging the heart valves permanently. It's a clear case where an antimicrobial response directly cross-reacts with self, causing autoimmune damage. Molecular mimicry really highlights how small sequence similarities can accidentally bridge the gap between fighting infection and attacking self.
Okay, so infection can break anergy via bystander activation, or it can directly trigger cross-reactivity via mimicry. Are there other environmental factors besides infections?
Definitely. We already touched on tissue damage itself. Significant injury from any cause can not only trigger inflammation and activate APCs, like in mechanism A, but it can also lead to the release of those sequestered antigens we discussed.
Right, from immune-privileged sites.
Yes. But also, damage can chemically modify normal self-antigens, creating what are sometimes called neoantigens.
Neoantigen, new antigens.
Essentially, yes. Self-proteins that have been altered maybe by inflammation, by enzymatic cleavage, or by chemical modification. The immune system is generally tolerant to unmodified self. But if a self-protein gets significantly changed, it might look foreign enough to trigger a response because the tolerance mechanisms never screened against that specific altered form.
Can you give an example of that?
Smoking is a really interesting environmental risk factor, particularly for rheumatoid arthritis. One hypothesis is that chemicals in tobacco smoke can get into the lungs and chemically modify certain self-proteins there, maybe through a process called citrullination.
Citrullination.
It's the conversion of the amino acid arginine to citrulline. This changes the protein's structure and charge. It's thought that these newly citrullinated proteins might be recognized as foreign by the immune system in genetically susceptible individuals, contributing to the development of RA, where anti-citrullinated protein antibodies are a hallmark.
So smoking might create neoantigens that trigger the disease? What about UV light? You mentioned that earlier too, especially for lupus.
Yes, ultraviolet radiation from sunlight is a well-known trigger or exacerbating factor for SLE. The mechanism likely involves UV light causing damage and death to skin cells, keratinocytes.
Apoptosis again.
Widespread apoptosis, yes. This leads to the release of large quantities of nuclear material from the dying cells, things like DNA, RNA, and associated proteins.
And normally the body cleans that up quietly.
Normally, yes. But in susceptible individuals, perhaps those with defects in clearing apoptotic debris or those whose immune cells are hyperresponsive, this flood of self-nuclear material might be problematic.
How so?
Because our innate immune system actually has receptors designed to detect microbial nucleic acids like bacterial DNA or viral RNA. Toll-like receptors, or TLRs, are key players here.
So the body mistakes its own DNA for invader DNA.
That's the idea. The excessive release of self-DNA and RNA after UV exposure might get inappropriately recognized by TLRs, like TLR9 for DNA, TLR7 for RNA, in immune cells like dendritic cells. This recognition triggers strong inflammatory signals, promotes the production of anti-nuclear antibodies, a hallmark of SLU, and generally fuels the autoimmune fire. So UV acts by causing damage that exposes self-antigens in an inflammatory context.
Okay, that makes sense. And one last factor often mentioned, especially for diseases like lupus, is gender.
Ah, yes, the gender bias. It's undeniable. Many autoimmune diseases, perhaps most, are significantly more common in women than in men. SLE, for instance, can have a female-to-male ratio as high as 9:1.
Why is that? What's the current thinking?
It's complex and not fully understood, but likely involves several factors. Sex hormones are clearly implicated. Estrogen, in particular, is generally thought to enhance immune responses, potentially tipping the balance towards autoimmunity in susceptible females. Testosterone, conversely, tends to be more immunosuppressive.
So hormonal differences matter.
They almost certainly do. Additionally, there are genes on the X chromosome. Since females have 2 X chromosomes, while males have 1 X and 1 Y, they have double the dose of X-linked genes. Some of these genes are involved in immune function, like TLR7 actually, and differences in their expression or regulation or issues with X chromosome inactivation might contribute to the increased risk in females. It's an active area of research, but the gender bias is a major feature of autoimmunity.
Okay, we've built this picture. Tolerance is complex, involving central and peripheral mechanisms. Autoimmunity arises from a combination of genetic susceptibility affecting those mechanisms and environmental triggers lighting the fuse. Now, let's maybe turn towards how understanding these fundamental biological pathways has actually given us proof points, both from rare diseases and even leading to some pretty spectacular breakthroughs in clinical medicine. Hashtag 3.1. Lessons from monogenic autoimmunity, proving the pathways.
Right. Those rare single-gene or Mendelian autoimmune disorders are just invaluable research tools, really, because they provide definitive human-level proof that disrupting just one specific component of that intricate tolerance network we described is actually sufficient to cause severe disease.
They isolate the variable, in a sense.
Exactly. We already talked about the AIR gene mutation leading to autoimmune polyendocrine syndrome, proof that central tolerance against peripheral antigens is critical.
And the FOXP3 mutation causing IPEX syndrome.
Proving the absolute necessity of functional TREGs for peripheral tolerance. We also mentioned FAS and FASL mutations causing ALPS, demonstrating the importance of apoptosis in deleting self-reactive cells.
So these rare, often tragic experiments of nature really confirm that the individual pathways we've discussed, AIR, FOXP3, FAS, are not just theoretical concepts. They're absolutely essential pillars holding up self-tolerance. Take out one pillar, and the whole structure can collapse.
Precisely. And they confirm that the complex, multigenic nature of common autoimmune diseases likely involves subtle defects in several of these pathways simultaneously, rather than one single catastrophic failure. But the monogenic diseases show us the core importance of each component.
Are there other examples from single gene defects?
Yeah.
Maybe involving B cells?
Yes, absolutely. For instance, there's a critical negative feedback loop for B cells. When antibodies, specifically IgG, bind to their target antigen, forming immune complexes, these complexes can bind back to a specific receptor on the B cell surface called FcRIIB.
Okay, an Fc receptor that binds the tail of the antibody.
Exactly. And when this FcRIIB receptor is engaged by these immune complexes, it delivers a strong inhibitory signal into the B cell, essentially telling it to stop making more antibody. It's a self-limiting mechanism.
Makes sense. Once you have enough antibody bound to the target, you don't need to keep churning out more.
Right. Now, if you look at mouse models where this specific FD receptor, FcRIIB, is genetically knocked out.
Let me guess, runaway antibody production.
Unchecked antibody production and spontaneous systemic autoimmunity, often resembling lupus. And polymorphisms in the human FEC gene that reduce its inhibitory function are also associated with increased risk for SLE.
So it confirms that even subtle defects in these normal negative regulatory feedback loops can significantly contribute to the failure of tolerance and the persistence of autoimmune responses we see in the more common complex diseases. Hashtag 3.2, clinical application of tolerance mechanisms. Okay, so the basic science, often informed by these rare diseases, builds the foundation. Now, perhaps the biggest, most high-profile clinical story to come out of understanding peripheral tolerance mechanisms recently is the whole field of immune checkpoint blockade in cancer therapy.
Oh, absolutely. It's been revolutionary. And it's the ultimate, perhaps slightly terrifying clinical proof of concept for the power of peripheral tolerance mechanisms.
How so? It seems paradoxical using tolerance mechanisms to fight cancer.
Well, it's about blocking the tolerance mechanisms. Remember those inhibitory receptors we talked about, the immune system's brakes, CTLA-4 and PD-1?
Yeah, the ones that normally shut down T-cell responses to prevent autoimmunity.
Exactly. It turns out that many tumors are incredibly clever. They exploit these very same natural braking pathways to protect themselves from being attacked and destroyed by the patient's own immune system.
How do they do that?
One common way is by expressing PD-L1, the ligand for PD-1, right on the tumor cell surface. When an anti-tumor T cell approaches and its PD-1 receptor engages with the tumor's PD-L1,
It gets the off signal.
it gets the off signal.
Yeah.
The T cell is effectively shut down or becomes exhausted right at the tumor site. The tumor essentially hijacks the body's own tolerance system, waving a "don't attack me, I'm self-flag" to evade immune destruction.
That's diabolical. So checkpoint blockade therapy.
The genius of checkpoint blockade is using therapeutic antibodies that specifically target and block either the inhibitory receptor, like CTLA-4 or PD-1 on the T cell, or the ligand, like PD-L1 on the tumor cell.
So you block the off signal.
You block the off signal. You prevent that inhibitory handshake from happening. By doing this, you essentially remove the brakes from the anti-tumor T cells, allowing them, or unleashing them, to recognize and attack the cancer cells much more effectively.
You're intentionally dismantling a key part of peripheral tolerance, at least temporarily or locally, to fight the cancer.
Precisely. And the dramatic success of these therapies in treating various cancers, melanoma, lung cancer, kidney cancer, and many others, is just stunning proof of how powerful those natural inhibitory checkpoint pathways really are in normally suppressing T-cell responses. When you block them, the immune system can really roll back to life.
If you're intentionally removing the brakes that normally prevent autoimmunity, doesn't that come with risks?
Yes, that is the crucial trade-off and the direct downside we see clinically. The use of immune checkpoint inhibitors carries a significant risk of inducing immune-related adverse events, IRAEs.
Which are basically...
They are essentially forms of iatrogenic autoimmunity caused by the medical treatment itself. Because you're taking the brakes off the entire T-cell population, not just the anti-tumor ones, you risk unleashing pre-existing low-level self-reactivity.
So patients getting these cancer drugs can develop autoimmune side effects.
Very commonly. They can develop conditions that look exactly like the spontaneous autoimmune diseases we've been discussing. Autoimmune colitis, inflammation of the colon, thyroiditis, inflammation of the thyroid, dermatitis, skin inflammation, hypophysitis, inflammation of the pituitary gland, even myocarditis or type 1 diabetes in some cases.
Wow. So the treatment that saves them from cancer can trigger a different kind of immune attack.
It absolutely can. Managing these IRAEs is now a huge part of using checkpoint inhibitors effectively. It perfectly illustrates that fine line the immune system walks, and that we are now therapeutically manipulating between effective immunity against pathogens or cancer and the dangerous potential for self-destruction. Hashtag 3.3. The cycle of chronicity.
Okay, one last major concept. We know autoimmune diseases are rarely just a one-off event. They tend to become chronic, often progressive, battles that can last for years or decades. Why is that? Why doesn't the system just reset itself once an initial attack happens? Why does it spiral?
That's a really critical question about the nature of these diseases. There are several factors contributing to the chronicity. One of the central ideas is a process called epitope spreading.
Epitope spreading. What does that mean?
Okay, imagine the initial autoimmune response is triggered against just one specific self-antigen, or even just one part, an epitope of a self-protein. Let's say it's a protein in the joint lining in rheumatoid arthritis.
Okay, initial target identified.
That initial immune attack causes localized damage and inflammation in the joint. Now, what happens during tissue damage and inflammation?
Cells die, stuff gets released.
Exactly. That initial fire releases a whole bunch of other cellular components and self-antigens from the damaged joint tissue, maybe different proteins, DNA, fragments of cartilage, things that were previously sequestered inside cells or just weren't the initial target.
Okay, collateral damage releases new material.
Right. These newly released or exposed self-antigens are then scooped up by the activated APCs that are already present due to the ongoing inflammation. These APCs then present these new self-antigens to other lymphocytes.
Including potentially other self-reactive lymphocytes that weren't involved initially.
Precisely. So lymphocytes reactive to these secondary self-antigens now get activated, joining the fray. The immune response spreads from the initial epitope or antigen to target additional self-epitopes and antigens within the same tissue or even related molecules.
So it's like a localized fire that starts burning adjacent buildings because the heat and smoke reveal previously hidden flammable materials. The attack broadens over time.
That's a fantastic analogy. The immune system essentially recruits new targets into the battle as the disease progresses. This epitope spreading is thought to be a major reason why autoimmune diseases like SLE or multiple sclerosis tend to become more complex, involve more targets, and worsen over time. The autoimmune repertoire diversifies and expands.
Okay, epitope spreading drives chronicity. What else keeps it going?
Well, you have to remember the immune system is intrinsically designed for escalation and persistence when it encounters something it perceives as a threat. It has built-in amplification loops.
Positive feedback loops.
Yes. Think about a normal infection. When T cells get activated, they produce cytokines that activate more T cells and also activate macrophages. Activated macrophages release cytokines that further activate T cells. B cells get help from T cells to make antibodies, which then help T cells and macrophages target the pathogen more effectively. It's a whole series of interconnected positive feedback loops designed to rapidly and massively amplify the response to eradicate the pathogen quickly.
But when the target is
When that same powerful amplification machinery is misdirected against self-tissue, those loops don't lead to clearance. They lead to more damage, which releases more self-antigen, which further fuels the inflammation and activates more immune cells. It becomes a vicious cycle. The system's own design for effective immunity tragically ensures that autoimmune injury is often not self-limiting, but rather prolonged, exacerbated, and locked into a state of chronic inflammation.
the system's strengths become its weaknesses in this context.
Perfectly put. And finally, thinking about the clinical picture, what ultimately determines the specific type of autoimmune disease someone gets? If multiple people have genetic susceptibility, why does one end up with psoriasis, skin plaques, another with lupus nephritis, kidney failure, and another with MS, neurologic deficits? That comes down to the dominant immune response that breaks tolerance. The specific arm of the immune system that fails and the nature of the target antigen really dictates the pathology.
Can you elaborate?
Sure. If the primary failure involves the production of high levels of autoantibodies that form immune complexes, antibody bound to antigen, which then deposit in tissues like the kidneys, joints, or blood vessels.
That sounds like type 3 hypersensitivity.
Exactly. That's the mechanism driving much of the pathology in diseases like systemic lupus erythematosus, leading to the characteristic nephritis, arthritis, and vasculitis. It's an antibody immune complex mediated disease.
Okay. What if antibodies aren't the main problem?
Conversely, if the dominant failure involved the abnormal activation of specific types of T cells, particularly Th1 and Th17 cells, which are potent drivers of cellular inflammation,
Then you get a different kind of damage.
then you get chronic inflammation and tissue damage mediated primarily by those T cells and the inflammatory cytokines they release, like interferon gamma from Th1 or IL-17 from Th17. This T-cell-mediated pathology is characteristic of diseases like psoriasis, multiple sclerosis, rheumatoid arthritis, which has both T-cell and antibody components, and some forms of inflammatory bowel disease.
So the specific flavor of immune failure, whether it's mostly antibodies or mostly Th1 cells or Th17 cells combined with the location of the target antigen, determines the clinical presentation and the name we give the disease.
Precisely. Understanding the dominant pathway is becoming increasingly important for developing targeted therapies too. #tag conclusion.
Wow. Okay, so this deep dive has really underscored that self-tolerance isn't just a passive state, it's an actively maintained process. It's a marvel of biological engineering, really, maintained through these sophisticated, multi-layered systems.
Absolutely. From the central screening in the thymus and bone marrow, heavily reliant on things like air and receptor editing,
To the continuous peripheral surveillance involving energy induction, the critical inhibitory checkpoints like CTLA4 and PD-1, and the active suppression by those FOXP3-driven regulatory T cells. It's incredibly complex.
And we now have a much clearer picture, though still incomplete, that pathological autoimmunity isn't just bad luck. It seems to be this catastrophic synergy.
Right, that combination of underlying genetic susceptibility, which
Yeah.
might weaken one or more of those tolerance systems.
And then an environmental trigger, maybe an infection,
Mhm.
significant tissue injury, UV exposure, perhaps even changes in our microbiome that comes along, activates local APCs, provides that inflammatory context.
It essentially unleashes those potentially self-reactive lymphocytes
Mhm.
that were either dormant or inadequately controlled, leading to chronic tissue destruction.
That model, the synergy model, like in figure 6.22 of the source material, really seems to capture the essence. You generally need both the genetic failure of tolerance and the environmental activation to really kick off and sustain a self-attack.
It takes both hits.
Seems that way for most common autoimmune diseases.
Which brings us back maybe to that final provocative thought, circling back to the cancer immunotherapy story.
Yeah.
We've learned so much about the body's own powerful immune brakes, CTLA-4, PD-1, that we can now therapeutically release those brakes to fight cancer. We are intentionally enhancing the immune system's power to attack.
It's a game changer for oncology.
But the critical question, the tightrope walk you mentioned, remains. If we become ever more skilled at boosting the immune system to fight tumors, how do we do that precisely without inadvertently triggering or accelerating that same unleashed system to attack healthy cell tissues?
That's the million-dollar question, isn't it? How do we target the enhancement just to the tumor microenvironment? Or how do we predict who is most at risk for IRIEs? Managing that delicate balance, achieving potent anti-tumor immunity while maintaining essential self-tolerance is truly one of the greatest and certainly one of the most urgent challenges facing medicine and immunology today.
A profound challenge indeed. Lots to think about there.